Bună ziua! În următoarele momente se va intona imnul României.
The Romanian anthem will be played in the following moments.
Domnule rector, stimați invitați, onorată asistență, dragi studenți!
Esteemed Mr. Rector, honored guest, distinguished audience, esteemed students.
Universitatea de Științele Vieții, Regele Mihai I din Timișoara, prin misiunea sa, promovează diseminarea rezultatelor cercetării științifice, a inovării și a transferului tehnologic către comunitatea științifică, academică și studențească, dar și pentru mediul economic, ca fundament pentru generarea cunoașterii și dezvoltarea societății.
The University of Life Science King Mihai I from Timișoara, through its mission, promotes the dissemination of scientific research results, innovation and technology transfer to the scientific, academic and student community, but also to the economic environment, as a basis for knowledge generation and society development.
În acest sens, Conferința Multidisciplinară privind dezvoltarea durabilă, organizată de universitatea noastră, oferă o oportunitate excelentă pentru cercetători, cadre didactice, specialiști din industrie și factori de decizie de a împărtăși rezultate și experiențe ale cercetării în domeniul științelor vieții, cu importante contribuții la păstrarea sănătății mediului înconjurător și asigurării unei hrane nutritive și sigure pe masa consumatorului.
In this respect, the Multidisciplinary Conference on Sustainable Development, organized by our university, provides an excellent opportunity for research, academics, industry specialties, and stakeholders to share results and experience on life science research, with important contribution to preserving environment, health, and ensuring nutritious and safe food on the customer's table.
Este a 7-a ediție a acestui eveniment de prestigiu, care reunește cercetători din mai multe zone ale lumii.
This is the 7th edition of this prestigious event, which brings together researchers from many parts of the world.
Pentru început, îl invităm pe domnul profesor universitar doctor Cosmin Alin Popescu, rectorul universității noastre, să adreseze mesajul de bun venit al domniei sale.
First of all, we invite Professor Cosmin Alin Popescu, rector of our university, to deliver his welcome message.
Da, mulțumesc frumos. În primul rând, stimate doamne, stimați domni, doamnelor, domnilor profesori, cercetători, dragi studenți, aș dori să vă spun bun venit la această conferință multidisciplinară și vreau în primul rând să adresez câteva cuvinte în limba română și apoi vă rog să-mi permiteți să adresez câteva cuvinte și în limba engleză invitațiilor noștri de peste granițe, de peste mări și țări. First of all, I want to say hello to everybody, and I will start to speak a few words in Romanian language, and after I try to say some information about the conference and other idea about this conference for you, for our guests in our university. În primul rând, vreau să vă mulțumesc frumos că v-ați făcut timp să vă opriți puțin din agitația aceasta pe care o simțim toți și, dragii mei, e important că ne facem timp și pentru astfel de lucruri. Eu, personal, probabil la tineri e puțin diferit acest sentiment de trecere a timpului, dar nu îmi vine să cred că suntem deja în luna aproape iunie. Sunt foarte prins ca și dumneavoastră și un cercetător, un om de știință, un om de educație, așa cum suntem noi, în mod special cei pe care am în fața mea și cei care sunt cu noi online. Înțelegeți lupta cu timpul și atunci când suntem implicați într-un subiect al cercetării sau într-un subiect al concepției științifice, artistice, și aici vorbim în mod special de această concepție științifică, ne luptăm indiferent de oră, fie că suntem dimineața sau după masa la birou sau după masa sau seara acasă, ne luptăm cu ideile și timpul pentru noi cumva are o altă formă, diferit cu cel al unei persoane care este implicată într-un proces, într-un ciclu de producție foarte strict și foarte bine definit. Este interesant în agenda unui om ca mine, ca dumneavoastră, cei care sunteți aici sau cei care ne ascultați, sunteți cu noi în mediul online, este interesant să-ți faci timp o zi, două, să pleci de acasă, să te organizezi, lăsându-ți cumva treburile, dar totodată în același timp le iei cu tine. Este interesant acest lucru, pot să apreciez foarte mult faptul că ne găsim timp să facem astfel de întâlniri. Este important că ați venit la noi în universitate, în Universitatea de Științele Vieții Regele Mihai I din Timișoara, nu pentru faptul neapărat că ați venit să vă prezentați ideile dumneavoastră, ci este important pentru faptul că v-ați făcut timp să vă întâlniți cu colegii mei, cu colegi care vin din alte țări, din Europa, din Africa, din Asia, din Statele Unite, sunt aici împreună cu noi, din alte universități, cred că peste 60 de universități, dacă nu greșesc, domnule, 80 de universități, din România și din afara țării. Câștigul mare pe care vreau să vi-l spun în mod special celor tineri, cei care sunteți de vârsta mea, oameni maturi, nu că tinerii n-ar fi maturi, dar câștigul mare după astfel de experiență este faptul că am putut să construiesc relații sănătoase, de prietenie sau de colaborare, care mi-au permis să mă dezvolt ca și om și ca profesionist. De ce spun acest lucru? Este pentru faptul că să ne uităm peste rezultatele cercetărilor noastre, să le dezbatem, dar nu aș vrea să dezbatem neapărat foarte mult pe anumite teme, ci aș vrea foarte mult să ne gândim cum putem să dezvoltăm relațiile dintre noi, din punct de vedere profesional, din punct de vedere științific, din punct de vedere instituțional. Vă spun asta pentru că cei care sunt aici, colegii mei din universitate, și cunosc foarte bine, sunt oameni pragmatici, deschiși, optimiști și cu dorința de a dezvolta relații naționale și internaționale. Sunteți într-un oraș de anvergură internațională, într-un oraș cosmopolit, într-un oraș intercultural, într-un oraș multietnic, într-un oraș care a trecut prin experiența capitalei culturale europene. Sunteți într-o universitate internațională în care conviețuiesc și muncesc deopotrivă români și alte naționalități, dar în mod special suntem plăcut copleșiți de prezența tinerilor studenți și cercetători din peste 30 de țări prezenți în universitate, care studiază în limba română, în limba engleză sau în limba franceză la diferite programe de licență și master. Provocarea pentru noi ca universitate, și este o invitație din partea mea pentru dumneavoastră, este aceea de a dezvolta relații în zona cercetării avansate, în zona cercetării aplicate, cu precădere pe ceea ce înseamnă zona de școală doctorală, unde suntem într-o efervescență a unei, era să spun, explozii, dar știți cum, când spun explozie, mă gândesc acum, pentru că suntem la Universitatea de Științele Vieții, la acea pornire a unui mugure care iasă din zona de protecție și dacă o punem așa pe rapid, chiar seamănă cu o explozie și toți cred că ați văzut modul în care iasă să vadă lumina soarelui acel mugure. Dar universitatea noastră de 80 de ani a dezvoltat educație, dar să știți că pe parcursul acestor 80 de ani, anul viitor, vom avea 8 decenii de existență. Ceea ce este important, ne-am adaptat la ceea ce înseamnă schimbarea, ce înseamnă schimbarea economică, socială, ideologică, și în mod special la provocarea la care facem parte acum. În mod special pentru generația tânără, ca un om care înțelege puțin mai bine decât generația copilului meu sau generația studenților noștri, înțeleg contextul global, internațional, nu în totalitate, dar îl înțeleg astfel încât să pot să iau decizii, să-mi formez o viziune și să construiesc anumite acțiuni împreună cu colegii mei din universitate pentru ceea ce urmează. Schimbările în știință, vreau să vă spun că în ultima perioadă, îndrăznesc să spun în ultimul deceniu, nu mă refer la 1953-54, da? După ce Watson și Crick au făcut o revoluție. Nu mă refer la 1956, când se vorbea pentru prima dată de inteligență artificială. Nu mă refer la 2022, când se vorbea pentru prima dată despre ChatGPT. Dar mă refer la această evoluție, la această explozie în care și un om educat, ca și mine, ca și dumneavoastră, este foarte greu să se adapteze. Intenționat am transmis colegilor mei și cu modestia care vreau să mă caracterizeze întotdeauna, vreau să spun și prietenilor care sunt acum în universitate sau în mediul online. Ceea ce am transmis despre reziliență la finalul anului trecut, începem să simțim acum. Și vestea bună sau vestea este că o să simțim pe măsură ce înaintăm ce înseamnă presiunea schimbărilor în zona științei, în zona academică, în zona în care noi activăm și poate o foarte mică parte dintre noi nu înțelege acest lucru, deși eu cred că toți cei care suntem aici înțelegem cât este de important să ținem pasul cu schimbările. Am ajuns la concluzia de foarte mult timp și la nivel personal, de foarte mult timp, și universitatea noastră știe acest lucru înainte de a înțelege eu acest aspect, faptul că împreună și izolați nu putem face nimic. Rugămintea mea este următoarea. Dacă îmi permiteți, vă rog frumos să înțelegeți că este important și se leagă foarte bine faptul că vorbim despre dezvoltare sustenabilă într-un context al multidisciplinarității. Adevăratele cercetări au împreună și adună un sociolog, un economist, un filolog, un profesor de matematică, un profesor de sport, un profesor de biologie, un profesor de statistică, un foarte bun specialist în zona de IT, un foarte bun specialist în zona de inteligență artificială, un foarte bun specialist pe zona de ce vreți dumneavoastră pe GIS și pot să continui cu exemplele. Aș vrea ca aceste diferențe și aceste bariere de pregătire educațională, și știu că de mult au dispărut. Noi personal avem o singură barieră, cea a respectului, și în momentul în care există bariera care ne oprește legată de respect, atunci se oprește și comunicarea, pentru că practic respectul incubă foarte multe lucruri. Dar aici, în această instituție, și vorbesc în numele colegilor mei, vă veți bucura de respect și de apreciere. Vă invit să deschidem colaborări în zona internațională. Pe lângă educație, credem puternic în internaționalizare în care am investit și investim resursă umană și resurse financiare și resurse de timp. Credem puternic în ceea ce înseamnă partea de cercetare, de inovare și de transfer tehnologic. Vă invit să deschidem proiecte împreună. Din experiențele pe care le avem cu partenerii noștri din țară și din străinătate, până la momentul acesta nu am trântit, cu ghilimele de rigoare, sau nu am avut experiențe neplăcute pentru niciun proiect. Nu o luați ca o laudă, luați ca un element al unei responsabilități în ceea ce ne privește. Mai mult decât atât, am înțeles ceea ce înseamnă importanța transferului către economia reală și în acest sens vă rugăm să luați aminte la un alt eveniment care va avea loc peste aproximativ 2 săptămâni și dacă mai este timp, parte din cercetările dumneavoastră, din brevetele, din zona dumneavoastră de invenții, ne-am bucurat să se regăsească în curtea unui eveniment, și când spun curte, este mic ca și zonă delimitată, mai exact în Salonul Internațional de Invenție de Inovație Traian Vuia, gestionat de universitatea noastră. Deci vă rog foarte frumos să vă uitați la aceste lucruri și suntem o universitate deschisă, așa cum spuneam ieri într-un interviu. Acest salon, chiar dacă este gestionat de invenții, inovații, acest salon internațional, vreau să vă spun că nu este doar al universității noastre, este al comunității. Faptul că îl gestionăm noi este un context, eu zic, favorabil și știind preocupările noastre, cred că este pe mâini bune. Vă invit și în acest sens și deschidem această oportunitate și pentru dumneavoastră. În cercetare nu are ce căuta egoismul, în cercetare nu are ce căuta lenea, în cercetare nu are ce căuta lipsa de răbdare, în cercetare nu are ce căuta acel orgoliu de a-ți învineți pieptul, în cercetare are loc foarte multă muncă, sacrificiu, respect, implicare și în cercetare cele mai bune rezultate, să știți că se fac atunci când suntem împreună. Cele mai multe idei, evident, nu trebuie să mă luați pe mine ca exemplu, dar cele mai multe idei într-o instituție ca și a noastră, vreau să vă spun că nu vin de la un singur om, adică mă refer la mine. Cele mai multe idei pe care dumneavoastră le veți vedea implementate într-o universitate ca a noastră, vin din partea echipei formate din aproape 300 de oameni, dascăli și cercetători, și mă bucur că este așa. Mă bucur că este așa. Aici veți regăsi cu siguranță un model de echipă, un model de responsabilitate, un model de altruism și spun asta nu pentru a scoate pe cineva sau ceva în evidență, ci pentru a vă încuraja să veniți alături de noi. Domnule prorector, Calman Imre, vreau să vă felicit pe dumneavoastră și echipa cu care ați lucrat. Vreau totodată să felicit colegii noastre prorectori care s-au implicat alături de dumneavoastră în a face o anumită regie și scenariu a ceea ce se întâmplă astăzi aici într-o formă constructivă și academică și apreciez această colaborare cu domnul prorector Iancu, cu domnul prorector Mergheș. Domnul prorector Stanciu regrete că nu este aici, s-ar fi simțit foarte bine în acest mediu internațional, veritabil, se reprezintă Universitatea în Statele Unite și deopotrivă mulțumesc domnilor decani prezenți aici și președintelui Senatului Universității noastre pentru implicare și pentru faptul că ați făcut posibil acest eveniment. Ca de fiecare dată vin și trebuie să spun doar câteva cuvinte, neștiind ceea ce urmează, neștiind cât ați muncit și e o situație puțin interesantă pentru mine, dar să știți că mi este confortabil și să vă țină Dumnezeu năravul. Mulțumesc foarte mult, stimați colegi, pentru ceea ce ați făcut posibil să se întâmple astăzi și mâine pentru cei care sunt prezenți în universitate. I'll try to say a few words in English. I don't know if I can repeat what I tell in Romanian language, but I want to express my joy. I'm glad because you are here in our university, Life Science King Michael I University, because I explain in Romanian language. Our university and our team, it's very open to work together with you, but I want to cover my words not only with words. I want to cover my speech with facts, and I want to explain you for people which is here for first time in our university and maybe first time in Timișoara. You are now in a very and really international city, multicultural city, multi-ethnic city, and the last year we passed the Capital Cultural European Capital Cultural. It was very interesting year for Timișoara. I invite to visit many buildings, many square, many interesting places, parks. You can visit, you can see interesting places in our city, Timișoara. In Timișoara, we have four public university, but the most I think interesting university, I'm sure it is here for you. But it's a little joke, but for me it's the most important university from Timișoara. It's normally. In the same time, you have a very, very good relationship with the West University, with the Polytechnic University, and the Medicine and Pharmacy University from Timișoara. I regard yesterday with our Vice Rector the program for today and for tomorrow. It was very interesting, the topics regarding your presentation, posters or oral. All the topics it is inside of our multidisciplinary conference, but please, it's important for you and for us, if you can speak, if you have a time for speak about our future collaboration. Because you know very well, because you are here, it's very important to understanding in this moment in Romania and in Europe and other countries, you cannot develop the big project only one institute. Like a rector, I invite to be with us in the very interesting project for the future. Here you can find my colleague, altruist people and very open, like a mentality, and not only people, my colleague, and I invite you to speak especially about our future, about our institution, about you. And for us, like international university, because in our university now we have more 500 students from 33 countries, from the Europe, from Africa, from Asia, from United States. You understand very well the internationalization and research and innovation. It's very important for develop one institution, one person, and one country. For PhD program, we start one strategy for develop in field of internationalization. I invite to be together with us in this strategy. I think it's a strategy, win-win strategy. And we must find together the advantage for everyone. Please, it's important for you, it's important for us. Thank you very much for because you reserve your time in your agenda for being our university. Because I know, like me and like my colleague, you are a full agenda, and I appreciate because you make time for visit Timișoara, for visit our university. Thank you very much. And for today and for tomorrow, I wish very, very nice days, and please make time for you, visit and take on coffee, tea in Center City or other part in Timișoara. Thank you very much.
Vă mulțumim, domnule prorector.
Thank you, Mr. Rector.
În continuare, îi dăm cuvântul domnului conferențiar universitar, dr. Kalman Imre, prorectorul Universității responsabil cu cercetarea, inovarea și transferul tehnologic pentru deschiderea oficială a conferinței.
Next, we called upon the University Associate Professor, Dr. Kalman Imre, the University Vice Rector in charge of Research, Innovation and Technology Transfer for the official opening of this conference.
Dear Mr. Rector, invited speakers, distinguished guests, and participants. First of all, on behalf of the organizing committee, I want to express my gratitude towards you for being present at the 7th edition of the Multidisciplinary Conference on Sustainable Development. Thank you for the interest you have shown, and of course, for the logistical and scientific efforts invested in your participation at our annual scientific meeting. We are extremely grateful to all of those participants from 18 countries, including Romania, Albania, Algeria, Bulgaria, Croatia, France, England, Greece, Hungary, Italy, Ireland, Northern Ireland, Kenya, Poland, United States, and Zambia, who have submitted 478 abstracts of scientific papers, contributing significantly to our efforts for maintaining this tradition. Scientific conferences typically bring together experienced scientists and emerging ones, being a great way of testing new ideas and to expose research results of their hard work to a wider audience. Regarding the diversity of submissions, we pride ourselves on combining a tradition of research with an open approach to the latest innovation in the multidisciplinary field of life sciences, including agriculture, bioengineering, horticulture and forestry, food science and technology, management of sustainable rural development, as well as Western medicine. We are firmly convinced that the scientific quality and content of the presentations and the innovative approach of ideas will bring out new perspectives and will establish future research directions in our assimilation process to the Western world. Furthermore, the challenges posed by the implementation of sustainable development in academic institutions will open up new opportunities for collaboration between participants. Last but not least, we are confident that the conference will offer its participants high scientific impact lecture, knowledge exchange, and memorable personal experiences. From this moment, I officially declare the conference open, wishing you a very pleasant stay at our university and in Timișoara, and a productive and successful scientific meeting. Thank you.
Vă mulțumim, domnule prorector.
Thank you, Mr. Vice Rector.
Pentru prima prezentare în plen, în cadrul conferinței, o invităm pe doamna prorector a Universității București, profesor universitar doctor Carmen Chifiriuc, coordonatoarea activității de cercetare științifică în cadrul acestei universități și a secțiunii de cercetare al Universității din București.
For the first plenary presentation of this conference, we invite Professor Carmen Chifiriuc, Vice Rector of the University of Bucharest, who is the coordinator of the scientific research activity within the University of Bucharest End of the Life, Environmental and Earth Science section of the Research Institute of the University of Bucharest.
Cu doctorat în biologie, doamna prorector este expertă în microbiologie, fiind fascinată de diagnosticul fenotipic și molecular al tulpinelor patogene, de bacterii și fenotipului de antibiorezistență.
With a PhD in biology, she is an expert in microbiology and is fascinated by the phenotypic and molecular diagnosis of pathogenic strains of bacteria and the phenomenon of antibiotics resistance.
Doamna prorector, aveți cuvântul.
Madam Vice Rector, the word is yours.
Hello everyone. I will stay here because otherwise you cannot see me. As a Vice Rector for Research of the University of Bucharest, I convey the warm greetings of our Rector Marian Preda of the University of Bucharest to your university, and hopefully we will have more intense collaboration in the future. I'm coming from the Faculty of Biology. In the University of Bucharest, we have 19 faculties. I am teaching, and my group is teaching general and applied microbiology, immunology, and virology. And our research directions are related to investigation of antimicrobial resistance, with a highlight to for biofilms. Biofilms are, as we will see later, a very important form of adaptation and resilience to different harmful conditions, including antibiotics and other biocides. Also, in the evaluation of novel antimicrobial strategies, either based on new drugs or on natural compounds, evaluation of the microbiota by culture-dependent and independent methods, and you will hear a very nice presentation of my colleague Grațiela Grădișteanu in the afternoon on this subject, anti-infectious immunity and microbial ecology. And I'm coordinating the Research Institute of the University of Bucharest, trying to create a collaboration platform of the researchers from the 19 faculties of our university, but also with researchers from other universities of the country or abroad. And in this purpose, we are offering fellowships. You are invited to enter the website and see the fellowships offered for postdocs, for young researchers, senior researchers, and for visiting professors. And also we invite research groups willing to implement their research projects at the University of Bucharest. The outline of my presentation maybe seems a little bit extensive, but I preferred to make this presentation in such way that those interested on different subjects could look later on the presentation, and those interested for collaboration could see what we can do at the University of Bucharest at the Faculty of Biology. I will start by presenting the main actors of the host pathogen interaction, then the anti-infectious defense mechanisms, and starting from this, highlighting the possible biomarkers derived from here. Then we will pass to intercellular communication, which is important, and we can learn a lot from the microbial world and the way it communicates inside microbial world and with their hosts. And then I will talk a little bit about resistance versus virulence and our experience in developing and implementing novel methods for the rapid detection of AMR and novel experimental models for modeling the host microbe interactions. So I invite you in this intricate dance of host pathogen interactions. We are just scratching the complexity of these mechanisms and try to see how the understanding of this mechanism could help us in developing novel diagnosis and therapeutic tools. Infectious diseases, we all know that they are in top threats for global health, and I don't think that only for health, global threats from 2019 to 2023. And in this host pathogen relationship, in this battle, let's say, a lot of virulence factors, a lot of pathogenicity features, and a lot of components of the host are struggling. And the result of this battle is either elimination of the pathogen or infection and disease. So we have the infectious agent coming with its virulence potential, but the density is also very, very important. And we have the numerous mechanisms of immune response evasion, let's say like molecular mimetism, like antigenic variation, and many other mechanisms. And we have the host, and here the health condition is important, and we are not talking only about the host, the human organisms or animal organism. We are also talking about their microbiome and also the immune status. That is the subject of my presentation today. So the host barrier is composed on of three main components: natural physico-chemical resistance, innate immunity, and adaptive immunity. And all these three components are together making this barrier and anti-infectious host defense. In this battle, another, let's say, modern actor interferes, and these are the prosthetic devices. The request for prosthetic devices is huge, and the market for these prosthetic devices is increasing exponentially in the last years. Particularly but due to the increase in the expectancy life and also the progress in the medical field. But these prosthetic devices are offering niches for the development of microbial communities called biofilms. And these biofilms are exhibiting resistance to immune system components and also to any therapeutic agent that we can administer for treating an infection. And you can see here the steps of biofilm development, starting from planktonic bacteria, isolated bacteria that are very, very susceptible to different therapeutic agents, and then they are forming these microcolonies and these communities, these citadels that are very well protected by this matrix and by other mechanisms from the action of different immune effectors and antibiotics. Therefore, the MICs, probably many of you have heard about these parameters, minimal inhibitory concentrations for antibiotics are increasing maybe until I found this in literature, 4,000 times higher values in order to be able to kill the biofilms. But we cannot kill the biofilms because inside biofilms, a small portion of the bacterial population is turning into a pair-sister population that is capable to survive in the presence of antibiotic or the microbial agent, and also another portion of the population is entering in a latency, metabolic latency form. They are not dividing anymore. This is the difference between persister and latent bacteria. And these huge concentrations of antibiotics cannot be reached in vivo without having side effects. So we are getting now much closer to that natural barrier. We are not talking here about immune effectors. Natural barrier is composed of physical factors like variations of pH, humidity, or chemical compounds like hydrogen peroxide and other similar compounds, and also different anatomical features like peristaltism, like ciliary movements, and so on. And another component comes here, but I won't talk about this because my colleague Graziela will tell you more later. It's the microbiome. Microbiota keeping the gates of the main entrance points of pathogenic agents in our host body, which are the skin and the mucosa. Probably you know that 90% of the antigens, pathogenic antigens entering our body are directly eliminated by the macrophages which are present in all our tissues, and especially by Kupffer cells from our liver. So only a small portion of pathogens could pass this barrier. The importance of the microbiota is so huge, and the microbiota it's intensively studied in the present, speaking about resilience. And even there are this metaphora, Homo sapiens symbioticus, modernicus, and dysbioticus, and this is referring to symbiosis with our microbiota, or reversible change in the microbiota or irreversible change in the microbiota called dysbiosis. And this has a lot to do with resilience, because resilience is not coming back to the same state, but coming back to a balance, another type of balance after a stressor is selecting for some adaptive mechanisms. And now we are passing to anti-infectious defense mechanisms, the immune defenses mechanisms. And the pathogen could trigger a very rapid response, leading to pathogen elimination after its unspecific recognition. And this task is performed by what we call salt and malt, skin-associated lymphoid tissue and mucosa-associated lymphoid tissue. However, some of the pathogens could escape this short route and entering on a longer route, including specific recognition of what we call molecular patterns, pathogen patterns, which are recognized by different receptors, very, very important receptors, leading to the activation of an inflammatory response and pathogen elimination. But, however, for some very important human pathogens and animal pathogens, this way is not effective, and we are calling for the special troops, and these special forces are represented by the specific immune cells, B and T cells, that are which are directly or indirectly eliminating the pathogen after activating an important inflammatory response. So this is the time from the pathogen entry. You see the strictly pathogenic agents are normally triggering a specific immune response and specific immunological memory. So at the second meeting with the same pathogen, we will act more rapidly, more intensively, and more effectively. The anti-infectious immunity could be synthesized in these steps: antigen entry, antigen recognition, antigen processing, antigen presenting to these immune cells, and production of immune effectors. And depending on the nature of the pathogenic agent, viral, bacterial, fungal, protozoan or helminth, a humoral or a cellular or a mixed humoral, a mixed immune response is triggered by a lot of cells and a lot of molecules, phagocytic cells, the cells of the lymphoid innate immunity, T cells, B cells, natural killer, and other related cells, complement, and antibodies. But how can these cells be called at the site of aggression? And this here come the importance of the receptors. I was telling you that there are a lot of receptors recognizing those PAMs, pathogenic molecular patterns, profiles. And a class of these important effectors are the toll-like receptors. There are 10 until now, but we don't know the ligand for the TLR10, but we know the ligands: bacterial parasites, yeasts, viruses. You can see each type of molecular pattern is recognized by a specific toll-like receptor. And this message of the nature of the pathogenic agent is adapted by some molecules that are triggering the activation of cytoplasmic pathways, and these activated pathways are on their turn activating the expression of genes for cytokines, chemokines, and other categories of signals that are important for the activation and for the proliferation of a certain type of cell depending on the nature of the aggression. As I told you, the pathogen is recognized by these TLR receptors in this case, but there are other more, and it is processed by processing the antigen. The antigen-presenting cell are taking out the most immunogenic parts of the antigen called epitopes, and they are presenting these epitopes to the special forces of the immune system, which are the T cells. And the T cells are differentiating, are polarized functionally in different subpopulations depending on the nature of the aggression. And these subpopulations are producing different sets of cytokines, either pro-inflammatory or anti-inflammatory cytokines. And if these concentrations of these mediators are in good balance, so the body, the human body will be zen during infection. Maybe we don't know that we have an infection, but if there is this balance in the production of this chemokine or cytokines or other mediators, we have what is called the cytokine storm with very deleterious effects on different tissues of the body and leading even to high lethality rates. So this cytokine profiling is very, very important for the rapid diagnosis of a microbial or viral infection and for monitoring the treatment and adapting the treatment. Sometimes if the pro-inflammatory response is too aggressive, too intense, an anti-inflammatory drug should be added to the antimicrobial therapy. And I will pass very quickly through these slides only just to show you how complex are these interactions, and if an imbalance occurs somewhere, it is very difficult, if almost impossible, to restore the balance. This is important. In bacterial infections, the main effectors are represented by the phagocytes, and the phagocytes are activated by pro-inflammatory cytokines. In fungal infection, the same. We have macrophages, we have neutrophils, but also we have natural killer cells, which are the effectors of the innate lymphoid immunity. In viral infections, the things are changing. Here, the most effective response, immune response, is driven by the cytotoxic T cells that are killing the farms of making new viruses, the infected cells. But at the same point, this inflammation could be so high that an anti-inflammatory response should be triggered in order to avoid the complications of an aggressive inflammatory, hyperinflammatory response. In protozoan infections, the effectors are very similar to fungal infections, but in helminth infections, the things are changing. Here we are confronting with something big, something that cannot be engulfed, and we need all the help possible. And besides the classical phagocytes, neutrophils and macrophages, here the eosinophils and also the mastocytes help in eliminating the pathogen and destroying it by what we call exophagocytosis. And only eosinophils are capable of this process of exophagocytosis or surface phagocytosis. So the cytokines are very good biomarkers. They can be very easy to detect in a non-invasive sample, but however, very slow progress has been made in the last years in introducing these cytokines diagnosis in the current diagnosis algorithms for infectious diseases. Hopefully, we have methods, standard methods to detect these cytokines. As you can see in these images, practically I made a print screen from the commercial institutions providing these sets of multiplex ELISA to detect Th1, Th2, Th17, and Treg cytokines. These four subpopulations of T cells are very, very important, and they are making the difference between a normal immune response and an exaggerated or sub-efficient immune response. And they are called with very, very nice names. Th1 cells are the warriors. We need warriors to avoid the pathogens. Th2 are overprotector because they are stimulating an anti-inflammatory response. They are stopping the immune system. Th17, they are the most hyperinflammatory, and that's why they are called troublemakers. And Tregs T regulatory cells are called peacemakers, because without these Tregs, our tolerance to our own antigens could not be possible. And for finishing the immune response after the pathogenic agent was eliminated, we need these Tregs. We need to have them there activated, and detection of typically cytokines produced by Tregs are practically a measure of their activity. Also, maybe you heard about macrophages polarization. The macrophages could be polarized towards M1 macrophages and M2 macrophages. And all of these both of these subpopulations are equally important in the elimination of the pathogenic aggression. We need M1 macrophages 1 populations in the first step of the infection, because we need a pro-inflammatory environment. We need to call by these pro-inflammatory cytokines other cells, but at the end of the infectious process, we need M2 macrophages to heal the tissues and to eliminate the destroyed tissues locally. So we have also methods to detect the specific cytokines characterizing M1 and M2 macrophages. There are a lot of challenges in introducing new biomarkers in the diagnosis, and specifically referring to cytokines, we are talking here about a huge complexity, and one single cytokine cannot be associated specifically with a certain pathogen aggression. That's why we need these multiplex assays of cytokines, and we need specific statistical tools to analyze this data and to propose some thresholds and some ratios between the most important cytokines. They have a very short life. This is very important in the conservation of the homeostasis of the organism. And they are active in very low concentrations, nanograms or even picograms. So we need very, very sensitive methods for detecting these cytokines. And also there are a lot of commercial considerations, but also the lack of databases. For genes, we have a lot of databases, so we can compare our results. Here, these databases are lacking. Probably you know from the veterinary field the same problem we have with Malditov diagnosis, because for certain pathogens, for example, some fungal species, these databases are not updated, are not upgraded, and we cannot take advantage of this very rapid and cheap method to really achieve a very specific and very rapid diagnosis. However, the landscape of the biomarkers of bacterial and viral infections has expanded very, very much in the last years. Probably all of us know about erythrocyte sedimentation rate, about C-reactive protein, about procalcitonin. These are the classical, let's say, pro-inflammatory biomarkers, but you can see here a lot of other markers derived from the ratios of different cells, blood cells, or from the detection of important pro-inflammatory cytokines like interleukin 6, 8, interferon alpha, and TNF alpha. And also, sorry, I don't know what is happening here, in that category name should have been TLR or NDR receptors, which are those important receptors specifically recognizing the molecular pathogens. And also the landscape of biomarkers of bacterial infections has expanded, and we have here adhesion molecules, which are very important for the accumulation of macrophages and lymphocytes and other cells from the blood in the tissue, in the infected tissue. We have a lot of chemokines. You know this CC or C or CX3C. C comes from cysteine residues. In some proteins, there is one in other two. These two could be one next to each other or could be separated by other amino acid residues. And this is, we have tests for detecting these chemokines or for detecting the receptors, the ligands for these chemokines. And a long, very, very, very long list with less known biomarkers that have been derived from understanding the host pathogenic agent relationship. One of these biomarkers are the heat shock proteins. Irrespective to the stressor, either viral or bacterial or parasitic or fungal, the cells are destroyed. And once the cells are destroyed, the cytosolic proteins, called heat shock proteins, are released in the external medium. These are called what we call endogen antigen and endogenous antigens. And for these endogenous antigens, the T cells have not been very well instructed in the thymus to recognize them as self, because the probability of the meeting between these cytosolic antigens and the cells, the immune cells, is very low and must remain low. This will assure our homeostasis. But in the case of an infection, these heat shock proteins are released in the external medium, are coming into contact with the lymphocytes, and the specific antibodies against heat shock proteins are released. And in one of our studies, we have studied the association of these specific antibodies between against heat shock proteins of 60 and 70 kilodaltons in gram-positive, gram-negative, and fungal infections, and we have seen that higher level of these antibodies specific to heat shock proteins are associated with gram-negative sepsis. And hopefully we will see in the future, a threshold for this biomarker for the evaluation and for scoring these gram-negative infections, especially the invasive ones leading to sepsis. What about communication? I was telling earlier that the cells, the microbial cells, communicate each other and communicate with the host. And this communication system is mediated by small molecules which are different in fungi, in bacteria, in gram-positive, gram-negative bacteria, and they are called quorum sensing molecules. And the production of these quorum sensing molecules, as the name suggests, is dependent on the bacterial density. So imagine a pathogenic agent. Imagine a bacteria, Pseudomonas aeruginosa, entering in the human or animal body. The density, initial density, is very, very low. And the concentration of these molecules is also low. But bacteria start to multiply and their density is increasing. Why? Because these quorum sensing molecules are upregulating the genes encoding for adhesins. And the pathogenic agent adheres to our tissues, and in that way the natural barrier of infection cannot be efficient in eliminating the pathogen. And the density of the pathogen increases until reaching a certain density. And at this threshold density, the pathogen is changing the expression of its virulence potential, also mediated by the quorum sensing molecules. They don't need adhesins, they need invasins, they need toxins to invade the healthy tissues. And this communication systems called quorum sensing is mediated by by different systems, which are called with different names. The best studied are those in Pseudomonas aeruginosa as model for gram-negative infections, and Staphylococcus aureus as model for gram-positive infections. These are the models also very well studied in our department during 2 or 3 PhD theses. And what I would like to focus now is a little bit on the structure of these molecules. I've told you that they are very different for gram-positive or gram-negative strains, but look on their resemblance with different antibiotics. We know about linezolid, last resort antibiotic. Linezolid that is effective against gram-positives, but it resembles very much with the homoserine lactones, which are the signaling molecules of the gram-negative bacteria. That's why gram-negative bacteria can be adapted and use some enzymes they are using to interfere with the communication in gram-positives, and they could adapt these enzymes to destroy linezolid. And this could be, when I've seen these molecules, when it was introduced in Romania, I think in 2002, something like this, and I've seen this structure, I told myself in a few years, we will hear about resistance mechanisms mediated by lactonases. And now it is like this. One of the resistance mechanisms to linezolid is mediated by the lactonases, which are naturally produced as an adaptive mechanism, competitive mechanism by gram-positive bacteria. And the examples are continuing. Look to the color here, quinolones, which are similar with hydroxyquinolones, nitrofurans, which are similar with furanosyl boride, cyclic dipeptides, which are very much similar with novel dipeptides that are proposed as promising antimicrobials. And probably the resistance to these compounds will occur very rapidly after the introduction of these antibiotics on the market. Studying these quorum sensing mechanisms, the science people have proposed the antipathogenic strategy, antipathogenic or antivirulence strategy, instead of the classical antimicrobial microbicidal one. So we don't kill bacteria, because if we kill bacteria, the bacteria will adapt, because they mutate very quickly, as you know, and they transfer very generously the resistance mechanisms horizontally. But if we just inhibit the production of virulence factors or the regulation, the coordinated regulation of the virulence expression by the quorum sensing mechanisms, we will convert a pathogen into a commensal without exhibiting any pressure, selective pressure for resistance. Resistance could occur, but not so rapidly as we can see for microbicidal agents. And you will find here a list of these quorum sensing inhibitors or antivirulence inhibitors. You see the efflux pumps are a preferred target for these inhibitors, because efflux pumps, you know, are involved not only in multi-drug resistance, but also in the secretion in the external medium of those quorum sensing inhibitors. And this is important. And don't forget, some antibiotics, some available antibiotics are exhibiting also antivirulence potential at sub-inhibitory concentrations. And this is demonstrated for rifampicin, for linezolid, and for azithromycin. And we have also demonstrated in our previous studies that some natural compounds, for example, phenyllactic acid, extracted from different probiotics, also act at sub-inhibitory concentrations by inhibiting the expression of different virulence factors, and also the expression of virulence regulators. And also you see a lot of these effects, antivirulence effects, of different essential oils or other purified compounds of plant or microbial origin. We have a lot of publications in this field. This is one of the most productive field in our department in terms of the number of publications and the number of collaborations. And also we are targeting specifically the biofilms, and also we have demonstrated by phenotypic assays and by QRT-PCR that indeed the probiotic soluble factors are inhibiting the expression of quorum sensing genes, and the pathogen, the pathogen cannot communicate with the host, or the host will gain the battle, and the pathogen will be eliminated without opposing any selective pressure on bacteria to adapt by resistance, or without inducing this biosis in our microbiome or the animal microbiome. Here you can see how this probiotic soluble fractions are just inhibiting the result of the QRT-PCR of the quorum sensing inhibitors. And we have tried to propose some simple methods to assess this activity, this antivirulence, this antibiofilm activity for these compounds. And we have used as a model a natural biofilm, which is the dental plaque biofilm. Dental plaque is a very complex biofilm, and we have just cultivated the dental plaque on blood agar. It should be read here, on blood agar, in the presence of DMSO, which is the solvent that we are using in microbiology, and here in the presence of ooznic acid extracted from lichens. And you can see the hemolysis has completely disappeared. So this is a very, very simple indicator that ooznic acid is exhibiting antivirulence activity. And also this activity has been demonstrated by different biofilm assay models that we have in our lab. In our lab, we developed a lot of assays also for appreciating the antimicrobial activity by classical methods against planktonic cells, but also for evaluating different materials, different surfaces which are covered or are modified to resist to bacterial colonization and the bacterial biofilm development. And I won't insist now on the methods. You can have the presentation and you can see the methods we are using in our lab, and we are very open to collaborate. That those quorum sensing molecules are also communicating with our host. And here is a diagram from a very recent paper showing a lot of neuromediators which are produced not only by our microbiome. For being happy, we need our happy microbiome, but not by our microbiome, but also by some pathogenic bacteria, especially gram-negative bacteria. They could produce these neuromediators or precursors for neuromediators that could affect the balance of the entire body and the homeostasis of different systems of our body. This communication with the host has been also studied by us using the model that I was telling we have studied a lot, Pseudomonas aeruginosa, and the quorum sensing molecules produced by Pseudomonas aeruginosa, and we have seen that this Pseudomonas aeruginosa affects the secretory profile and the apoptosis rate in mesenchymal cells. And this is very, very important because especially in lung infections, in pulmonary lung infections with Pseudomonas, we see these transitions to epithelial to mesenchymal cells. And this transition is the way to fibrosis. And by simple assays like this, we can predict for a certain bacterial strain this potential for these complications. What about experimental models? We are searching for the right experimental model, but each experimental model, each of them has advantages and disadvantages. I won't enumerate these disadvantages. You can have the presentation. What I would like to tell you is that in our lab, we are working with Drosophila melanogaster. A lot of Nobel Prizes have been received for Drosophila melanogaster as a model for different diseases, including infectious diseases. And in our studies, we have demonstrated very clearly that the genetic predisposition of the host, gamma COP gene, this is a gene involved in the vesicular transport from endoplasmic reticulum to Golgi apparatus. And this is involved in a lot of immune processes. And this genetic predisposition of the host influence the resistance of the host to infection by Pseudomonas aeruginosa. And I will rapidly pass through these slides because resistance is part of virulence. Virulence and resistance are sharing the same genetic platforms, are a response to certain stressor, and the most important stressor is the high concentration of antibiotics used not in the clinical sector, but in veterinary sector. And yes, this resistance in our previous studies, we have clearly seen by sequencing the strains that resistance genes are flanked by biocide resistance genes. And this is an important finding, I think. And also what is important to see is that some specific, very important strains are are surviving in the hospital, but you can extrapolate to the veterinary field, are surviving the chlorination effect and are getting in the waters. And this could help us to propose some candidate biomarkers for controlling this phenomenon in different environments. And this is all for me because instead of conclusions, I just raise the questions, the awareness on the limitations on understanding this pathogen-host complex interrelationship, but this pathogen interrelationship could contribute essentially to improve the surveillance, the diagnosis, and the treatment of infectious diseases. Thank you very much for your attention.
Vă mulțumim, doamna prorector, pentru interesanta prezentare și pentru că ați ales să fiți alături de noi în această zi importantă pentru universitatea noastră.
Thank you, Madam Vice Rector, for your interesting presentation and for choosing to join us on this important day for our university.
A doua prezentare va fi susținută de către domnul dr. Petre Jitaru, cercetător în cadrul Agenției Naționale pentru Siguranța Alimentelor, Mediului și a Sănătății Ocupaționale, Departamentul Oligoelemente și Metale, Maison Alfo, Franța.
The second presentation will be given by Dr. Petre Jitaru, researcher at the National Agency of Food Safety, Environment and Occupational Health, Department of Trace Elements and Metals, Maison Alfort, France.
Dumnealui a finalizat studiile doctorale la Universitatea din Antwerp, Belgia, după care a parcurs mai multe stagii de cercetare în cadrul Consiliului Național de Cercetare din Veneția și în Laboratorul Național de Metrologie din Paris, iar în prezent, ca valoros om de știință și expert în chimia alimentară, conduce activitatea de cercetare în cadrul instituției de unde provine.
He completed his doctoral studies at the University of Antwerp, Belgium, after which he completed several research internships at the National Research Council in Venice and at the National Metrology Laboratory in Paris. and is currently as a valuable scientist and expert in food chemistry, leading research work at his home institution.
Domnule dr. Jitaru, aveți cuvântul.
Dr. Jitaru, you have the floor.
Bună ziua. Thanks for the very nice introduction. I didn't expect that. First of all, I would like to thank the organizers for inviting me today to this very nice city and very nice conference, and especially to Professor Ersilia Alexe, with who I hope to collaborate in the future. We wrote a project together. If the project will be accepted, I hope to come more often in Timișoara. That I start to like already, to love, because it's the second time. So, yeah, you had my introduction very quickly. I'm Petru Jitaru. I am the head of the laboratory dealing with trace elements and minerals, and it will change the name in a few days. Actually, it's trace elements and nanomaterials. My work is very technical, of course, because I'm a chemist, analytical chemist, but today I'm going to speak something a little bit more general, and I will present the food safety assessment at national scale, and I will share a little bit of our French experience. Because, you know, there is a very good French cuisine, and very good French cuisine, in fact, requires a very good food safety. So here you can see a few examples of different types of chemical analysis related to food. We can make chemical analysis in terms of quality control, in terms of nutritional studies, in terms of biomedical studies, authenticity and fraud. But what is most important, especially related to my work, is in terms of risk assessment. So what is risk assessment? So I would like to give you an example, because sometimes there is a confusion between hazard, or we can call it danger, and risk. So actually the hazard or the danger is a potential source of harm. For instance, the presence of a shark in a sea is a hazard, okay? So there is no problem if you don't swim together with a shark. So once you swim in the proximity of a shark, you expose yourself to a risk. So it's the same with the food. Actually, the risk is the likelihood that a hazard will occur and resulting in a harm. Of course, if the shark is small, the risk is small. If the shark is big, the risk is big. If the shark is very big, the risk is very big as well. So how we characterize the risk? So there is a whole science related to that. So I will be short and general. Actually, to characterize the risk, we have first to characterize the danger or the hazard, okay? And this involves chemical analysis, but also toxicological studies, because in fact, we have to correlate, I will show you a little bit later, we have to correlate the level of a contaminant or a hazard in a food item with its toxicological response. But it's not enough to make chemical analysis. We have also to know how much we intake, how much we eat that chemical hazard. Actually, how much food we eat, and that food, how much contains one or more chemical hazards, okay? So the combination of the two activities leads, of course, to a risk characterization. So very quickly, actually, the risk is characterized by what we call intake or daily intake, which can be also weekly intake and so on, which is calculated with this formula. Actually, is the product between the intake and the concentration of a given contaminant or a given hazard, depending on the body weight. That means we make the ratio to the body weight. So a general example, these numbers are for cadmium, but it's not important. Generally, what we do is to classify the foods into different families, okay? We analyze the food, but we need to know the consumption. As I said before, the risk is a product between the intake and the contamination. So we need to know how much of different type of food we eat. We transform this consumption or intake into gram per kilogram body weight. In this case, we divide it by a body weight of 60 kilos to obtain this number. And then we have the level or the concentration of a given contaminant in this type of food, and we calculate the exposure, which is the product, as I said. And then we have what we say cumulated exposure from different type of foods, and then we have a sum. Okay, this is the cumulated intake of, for instance, cadmium in a typical diet. And how we do with this number? I will not go into details, but this number is actually compared with what we call toxicological reference value. So if this number for the contaminant that we speak about is less than the toxicological reference value, that means there is no risk. If it is higher, there is a risk. So it's the equivalent with swimming next to a shark in the sea. So I would like to say a few words about the importance to know the intake of eventually contaminated food. And before that, I would like to summarize a little bit the idea of risk assessment. Actually, the idea is very simple. We can eat contaminated food. I don't say the level, but we can eat contaminated food if we don't eat too much. And also we can eat contaminated food. If we eat contaminated food but not too much, the risk may be limited. So if one type of food is contaminated, it doesn't mean directly there is a risk, because it's the intake that controls the risk in the end. And on the other hand, we can eat food that is very little contaminated, but if we eat a lot, we can have what we call a chronic risk. So there is eventually an impact on long term. So it's very important to characterize the food in terms of the danger, but it's extremely important to know what we call the diet habits of a population. And in France, actually, we take care a lot of this type of studies, and you see here we have already carried out the third, what we call INCA study, or National Individual Study for Food Consumption. I will not enter into details. To almost each slide there are some links and where you can find the reports. The reports are of course very big and so on, but you can find all the information almost that I speak about. Okay, so this National Individual Study for Food Consumption have different purposes, but one and the most important purpose is to know how much of different types of food is consumed, of course drinks and food supplements, and then there are a lot of other possible information that we can get. And here you see in a very, very simple way how the result of this kind of, in fact it's a survey, it's a questionnaire, it's not, there is no analysis involved in this case. So you can see here, for instance, we have data for infants, for children, for teenagers and for adults, and you can see there is a distribution of different types of food, okay, in what we call a daily diet. So this allows us to carry out the risk. Without this kind of information, we cannot evaluate if a food that we consumed is risky or not. So a few words about the food safety in France. I would like to say that I live in France for 16 years now, and I work in this field directly or indirectly, and there are some pictures there, but unfortunately we cannot see them, but I will speak. Actually, the food safety in France is one of the most efficient in the world. You can see here that every year, actually in France, we control almost 400,000 farms in terms of food safety, but in a very general way. Almost 700,000 establishments, all type, are controlled every year. Almost 6,000 people are dedicated for the food system in France. 14,000 veterinarians are also involved, and the budget for ensuring the food safety in France, the budget for a year, is almost 600 million per year. So the food safety is ensured by the Ministry of Agriculture, and more specifically by the French Directorate General for Food. Okay, most of the risk assessment is carried out at my work, actually not in my lab, but we have a dedicated department for that, which is called Risk Assessment Department. So a few words about my institution. Actually, what is what is interesting with ANSES, the institution where I work at, is the fact that the risk assessment, and not only, is done by external experts. So we are totally independent, totally transparent, and the government cannot control our institution at all. So to ensure the total transparency, everything is done by external experts. We have around 800 external experts that come regularly in different commissions at ANSES. And these experts are organized either in specialized expert committees, COS, either in working groups, which are all the time, or sometimes we have what we call collective emergency expertise groups, because sometimes there is really something urgent to answer, and in this case we have to provide an answer to the ministry in a few days and maximum a few weeks. So you see here the list of all these specialized expert committees. I will not enter into details, but the one that is interesting for me is the assessment of physico-chemical risk in food. Now, how we carried out the risk assessment? In fact, there are two main tools for doing that. The first tool, or the first approach, is based on monitoring and control plans. Monitoring and control plans actually are what we call official analysis. In each country, European country, we are obliged by the law, by the European law and also national law, to ensure official analysis. Each country puts in practice, eventually in a different way, this, but it's compulsory to have official analysis, and these are monitoring and control plans. Very quickly, monitoring plans involve a random but representative sampling of different food from a target population, and the control plans are targeted sampling. Okay, so that means we have a signal of a possible contamination of food, so we go specifically to identify a number or a family of chemical hazards in a selection of food items. And these monitoring and control plans focus on three types of dangers or hazards: chemical contaminants, biological contaminants like bacteria, viruses and parasites, but also physical contaminants in France, specifically on radionuclides. So I give you now the numbers related to the work around the monitoring and control plans in France. The last update that I have is from 2021. So in 2021, and I suppose later on also in the last years, we carried out 21 monitoring and control plans involving the determination of more than 200 chemicals and about 60,000 samples. So this is every year, 60,000 samples, and the budget only for this part, monitoring and control plans, is 13 million per year. And the conclusion of all of this is that all of these monitoring and control plans, so official analysis in France, less than 1% of the samples were what we call non-compliant. Because this official analysis, in fact, try to see if the levels of what we call regulated contaminants are below the maximal legal limit or the maximum admissible limit. So here you cannot see very well, but I gave some examples of substances which were not compliant, so like veterinary drugs, chlordecone, which is a specific French problem, and so on. So the idea of monitoring and control plans is a little bit interesting because we have the tendency to analyze almost everything. Okay, so we went in the last 20 years, to analyze a lot of stuff, a lot of food, which was not eventually necessary. I mean, we focused on chemicals which were not regulated, I mean, which were not imposed by the law. Also, we analyzed food items which were not eventually imposed by the law. So, of course, this is not sustainable because it costs a lot of money, a lot of time, and so on. So at my agency, actually the ministry asked ANSES, my agency, to make a study to see if all these monitoring and control plans are efficient and eventually sustainable. Of course, when we have a question like this raised by the government, it takes several years to answer. So, and it's a committee of experts that is answering this question. And here you see the report, which is very big, but here you have the conclusion of this report in a very simple way. Actually, try to see if we have to maintain, to reinforce or reduce eventually the number of analyses. And we did that for what we call regulated analyte matrix couples, I mean for the things that are imposed by the law, and also for the non-regulated, because we wanted to see if besides the regulated food items, there are eventually some risks, okay? And you see here that 74% of the regulated work that we did, or we do, should be maintained. But about 17% we have to reinforce. So even in terms of regulation, we have to reinforce this type of activity. And a little bit of this activity, legal activity, should be reduced, like about 9%. And the conclusion is totally different in case of what we call non-regulated analyte food matrices. And you see, we have to reduce about 66% of what we do because it's not necessary. Of course, we have to reinforce a little bit. We have to maintain just a little bit, but what is also interesting that is we have to create new activities, even for the non-regulated activities. So again, this is the conclusion a little bit more in detail, but it's very interesting to see what is necessary eventually to reinforce. Here you see the different type of food items, and here you see the chemicals, okay, the danger. And for instance, what is in red means we have to reinforce, and this can be, in my opinion, everything or almost everything that we find in France is available for Romania as well. So you see here, when we speak about trace elements, we have to reinforce this kind of controls for the alcoholic beverages. We have to reinforce also partially for vegetables, food based on vegetables. And if we look to the neoform compounds, like acrylamide, for instance, we have to reinforce this kind of activity for foods for infants, for children, okay? Also for cereals, also for snacks based on potatoes and so on, because this kind of neoform compounds like acrylamide result when we cook a lot the food, and so on. For instance, an interesting case is also perchlorate. We have to reinforce this kind of control in terms of fruits and also vegetables. And I will not enter details. So the same type of conclusion for the non-regulated situations, okay? So even if it is not regulated, so not imposed by European law, there are some situations where we have to take care. Again, the neoform substances should be reinforced for the food for the children. So you see that infant food comes regularly, so we have to take a lot of attention, to pay a lot of attention to the food for the children. Okay, so this was the first approach that is currently used for risk assessment, because we have this data. These data are available everywhere, in Romania, in France, and so on. So we have data coming from official analysis, and this allows us to carry out the risk assessment. But there is a second approach, which is based on total dye studies, and this approach is more scientifically oriented. So what is a total dye study? Unfortunately, if I'm not wrong, there is no any total dye study realized in Romania so far. So maybe there is the time to do that in Romania. Maybe we can help with that. So a total dye study is actually a study carried out at national scale, which focuses on the assessment of the contamination of a general population. It can be the general population or a specific population. So it's focusing on the contamination in real life conditions. So this means that we analyze food that is prepared, we say, as consumed, because the monitoring control plans that I just spoke about actually use raw food, for instance, raw meat, raw fish, raw powder, and so on. So there is no food, actually. So the total dye studies go further and ask the question, yes, maybe raw meat doesn't bring any risk, but what happens when we put different type of meats, different types of vegetables, different types of seed in the food, okay? So, these total diet studies are a step further in terms of risk assessment, and in France we have quite experience with this kind of studies, which are of course very costly and take a lot of time. We are now carrying out the 4th total diet study, so I'll present you very quickly what we did so far. So actually, to be representative, we have to select the food that is the most representative for instance, for Romanian population. So what does it mean? I will not enter too much in details. So that means we have to go to the regular supermarkets. So no to, I don't know if there are this kind of supermarkets, but not supermarkets for VIPs, for instance, but regular supermarkets. We have to take different type of food, for instance, fresh food, canned food, the same. For instance, we can have fresh fish, but canned fish. So we are supposed to not focus on a single brand, but to take as many brands possible. And what is interesting is that we have to prepare the food. For instance, we are not going to, in generally speaking, we prepare the food like it is consumed. For instance, there is a special laboratory working with us, which prepares different soups, prepare pizza, sandwiches, and so on. So we don't buy the sandwich from the market, because we need to prepare that in controlled conditions. Okay, otherwise we can have some biases. So we prepare the food as it is consumed by peeling it, washing, and so on. We eventually use different type of cookings, using frying pans, microwave, classical oven, and so on. Eventually, we can also select, if possible, half regular and organic food, and so on. So this is the image. This kind of study should be representative of a general population. So what we did in France so far, sorry for the colors, they are not very visible. Actually, we carried out the first total study in France in between 2001 and 2005. From our experience, a study like this takes 5 years, in general 5 years, and it starts with collecting the samples from practical point of view, collecting the samples, preparing them, shipping them to different labs, and then analyzing them, and there is a lot of work to really calculate the risk, which is done at the end, of course. So the French total diet study, you see here, it was a little study, I would say. We collected more than 2,300 different type of foods for that, and we focused on about 40 chemicals, and this led to 40,000 analytical results. When you multiply the number of chemicals with the number of samples being analyzed, you generate quite a big number of analytical results, and the budget at the time was about 1 million. And then we carried out a second total diet study 10 years later, more or less, where we wanted to go a little bit more in detail, and then we took more or less 20,000 different type of foods, and we focused on about 450 chemicals, which is a big number. And this led to 250,000 analytical results that needed to be treated at the end. And the budget was about 4 million. And then what is interesting, that we carried out a third total diet study, but which was focused on infants. So it was the last one, actually. So you see exactly the same strategy, but we took into account only food for children less than 3 years. And of course, we had already some experience. We had, because we speak about children, we took less samples this time, like more than 5,000 different type of samples, but we had a lot of chemicals, 670 chemicals being determined. And this led, of course, to a big number of analytical results. I will not enter into details. And now there is what we call the third total diet study, and my lab is involved with the analysis of this. It will hopefully end in next year. And here you see we collected only less than 9,000 food products compared to 20,000 food products. What does it mean? That means we got some experience, and we wanted to make this third total die study more sustainable. And also, instead of 445 chemicals, we have only 260 chemicals. We reduce also the number of samples, but the number of chemicals, because we have already a lot of experience and we focus really on the signals that we detected and we confirmed before. The budget of this project is now about 4 millions. So this I already said. Okay, we have to be always sustainable. So the tendency to go too much into details now is almost finished. So now we focus because we have some experience. Well, I will not enter to details, but these are the chemicals which are now investigated in the third total die study: trace elements, mycotoxins, contaminants from human activities, from food contact materials, pesticides residue, phytoestrogens, neoform contaminants, and others. Okay, so here you see the results. Sorry, the extract is in French, but here you see some, in a very simple way, which were the conclusion of the infant total die study, which was the last one. Okay, so we classified the risk into 4 categories. In red, it's a serious risk, I would say. In yellow, it's a medium risk. In green, there is no risk, and we have a 4th column where we cannot draw any conclusion, and I explain you why. For instance, if we focus only on trace elements, because there are too many details here, so there is a risk, so there is a worrying situations, as we say, in terms of lead, inorganic arsenic, and nickel, and we speak about food for children, less than 3 years, okay? But we cannot exclude the risk in terms of aluminum, in terms of methyl mercury, strontium, chromium 6, and so on. And luckily, there is no, at least in the previous study, there is no risk related to chromium 3, inorganic mercury, and antimony. And then there is a list of few elements for which we cannot draw any conclusion. Why? Because the data were not enough good. Generally speaking, if the result, for instance, is not quantitative, I mean, the level is low than the limit of quantification, it's very complicated to exploit this kind of result. Okay? So what is new for this third total die study? This is my topic. Actually, for the first time, we wanted to go a little bit beyond the trace elements, and then we focus on species of different elements. For instance, in terms of mercury, maybe you know, the species the most toxic is methyl mercury. So we are now focusing specifically on inorganic mercury and methyl mercury. In the same way, you know that arsenic is toxic. You all know maybe that arsenic is toxic, but there are some species of arsenic which are much more toxic than others. So this is the inorganic arsenic, and we are going now to focus on inorganic arsenic. And then also you may know that chromium is toxic, but not all the species. Chromium 3 is not toxic. Chromium 6 is very toxic. So we focus now on the 2 species of chromium, okay? So this is quite a hard work to do. So we have a PhD. I am directing a PhD project to develop the methods for that and to apply, of course, for the third total study. So which are the conclusions? Of course, the monitoring and control plans are a very good approach, a very good tool to carry out the risk assessment. And the advantages of the control plans is that we can have a temporal trend because we are forced to do that every year. So we can detect signals every year, okay? But the disadvantage of this official analysis is that they focus, as I said before, on raw samples. So is not the information is not complete. So to get the best information, we have to carry out total die studies, and the conclusion, at least for France, is that the total die studies that we carried out so far in France confirm the high level of risk management, of course, in terms of chemical contaminants. But as you saw just before in red, for some categories of population, there is some risk, so we put in evidence that there is a risk, and of course we have to deal with that risk. And this is the work of the authorities. And also you saw the 4th column, where we cannot conclude. Why? As I said before, either there is no toxicological information, so we can detect or we can quantify a contaminant in a food, but we have no idea how much toxic it is because we don't have yet what we call the toxicological reference value. But most importantly, we need to develop continuously, I would say, the best analytical techniques to be ready for such a study. So that's why at ANSES, at my institution, we have labs working continuously to improve the methods, okay, that can answer actually in this kind of studies. Thank you very much for your attention.
Vă mulțumim, domnule doctor, pentru valoroasa expunere și că ne-ați onorat cu prezența de la o distanță apreciabilă.
Thank you, Doctor, for your valuable presentation and for honoring us with your presence from an appreciable distance.
În continuare urmează prezentarea domnului cercetător științific Florin Mircea Iliescu, de profesie biolog și doctor în științe, cercetător în cadrul Institutului de Educație Continuă, Universitatea Cambridge din Marea Britanie, care va încheia sesiunea de comunicări din aulă.
Next, the presentation of Mr. Florin Mircea Iliescu, a research scientist and biologist, PhD researcher at the Institute of Continuing Education, University of Cambridge, UK. He will conclude the session of communication in the aula.
Un cercetător britanic veritabil, născut și crescut în București, dar cu doctorat la Cambridge, pasionat și fascinat de evoluția și de diversitatea umană.
A junior British researcher born and raised in Bucharest, but with a PhD in Cambridge, passionate and fascinated by human evolution and diversity.
Domnule doctor Iliescu, aveți cuvântul.
Dr. Iliescu, please have your say.
I know it's late and I know you're hungry probably, so I'll try to be quick. Thanks so much for the invitation, thanks to the organizers, thanks to Dorin Kamen for insisting for inviting me. It's very nice to be here in Timișoara again. It's nice to be at this university. I like the name. I think it's fitting that the name is of King Michael of Romania. I think it's a good name. I'll try to keep this. Yes, so my name is Mircea Iliescu and I'm a biologist, I'm a geneticist. I work mainly on humans, and I know that probably here most people don't work on humans, but I will try today to show that studying humans is quite relevant, and in a way it's a big connection between everything. That's why it's life sciences. And I think it's very interesting and important to think about our place in the world and our history and our connection with plants and animals. So that's what I'll try to show today, and also to discuss how technologies that we've been using for humans are being used and will be used a lot in studying all kinds of organisms, from bacteria to plants and animals. So if we talk about human genomics, the first draft of a human genome, which was basically reading the genes reading more than 90% of our genome, was finished in 2001, and the cost was around $3 billion. So $3 billion, and it took about 10 years. Now a human genome is being sequenced in a few hours, and the cost is at around $600. So only by saying that we realize the huge leap in technology. I think the rector, Mr. Rector, was talking about this huge leap in technology, and you can already imagine that this huge leap in technology has benefited the study of all the other plants and animals. So in a way it's interesting because we're humans and we're selfish and we really want to study ourselves, but by developing the genetic technologies for studying humans, we are now really good in studying everything really fast. Of particular interest is the idea that you can take everything from an environment, be it a lake or anything else, and you just look at everything, you sequence everything. Actually, even more interestingly now, we know that we leave traces of genes everywhere. There's been a study to show that, if you go on a beach, your DNA will be there. So you can basically just look in a place and see what animals, what plants have been around. So all this in a way has been possible because we've been developing this technology and because the cost is less. So I'm staying a little bit on this because this is crucial. In our field, in life sciences, if you want to do research, you need money. And the fact that now the costs of genetic sequencing have been decreasing so much means that you can actually do many things that you could not have done before. So the difference is from $1 billion, let's say, dollars to $600 for one human genome. So you can imagine that studying plant genomes or other genomes will be probably cheaper. But today I'm just going to tell you about humans. I'll tell you some things that I do or some things that I'm interested in. And then, of course, we can discuss about possible connections with many other things. So, okay, the lights are... Can we maybe try to turn off some of these lights, if possible? So, I'm a biologist, I'm a geneticist, but I've always been interested in, if we can look at genes, trying to use that rather than for medical purposes, but just to understand our history and our diversity. So I'm really interested in the idea of how can you use biology to understand our diversity and our history. So this is a very, yes, thank you, that's much better. This is a very schematic and broad map. This is the tale of our history and evolution in just a few lines. But basically, all these things here we know from archaeological evidence, but more recently from genetic evidence as well. So in other words, take many populations, you compare the genomes between them, you see how they split, you see how they mixed, and you can have this kind of map saying that humans mainly went out of Africa around 60,000 years ago, that they were in Australia by, oh yeah, the map is missing the continents, but yeah, sorry about that. That humans were already in Australia by 40,000 years ago, and they were in the Americas by 15,000 years ago. Interestingly, when we think about genetic diversity or genomic diversity, we know that most genomic diversity of humans is in Africa, and that as humans have been moving out, we call them serial founder effects. So in other words, you have a population and then a subset moves further and then another subset. We know that basically genetic diversity almost decreases in an almost straight line from Africa to the Americas. So the least diverse human populations would be Native Americans and the most diverse would be Africans. And this has a lot of implications for medical genetics. This is what us geneticists can do. So basically, each of these lines is an individual. So if you sequence a whole genome, then you can use very complicated statistical bioinformatic methods and start doing these kind of colors and to show how different populations are. So these would be some Africans, these would be other populations. So you can start seeing each individual as a product of different ancestry components. And this is relevant in any other organism as well. So every organism would be a product of different ancestries. As humans moved out, we know that they were rather quite fast. It's difficult to know the exact road because probably they were moving on the coast, and that coastal area now is underwater. But anyway, that was quite fast. When I say fast, I mean they were already in Australia by 50,000 years ago. And that means within 10,000 years, they did all the distance from Africa to Australia, which in those times, it's really fast. Okay, so my Mac computer has, this doesn't like my Mac computer. So sorry, some things will be missing, but I'll try to be very fun and nice and talk to you about this. This is what I was talking about, the serial founder effect. So you lose diversity as you move out, and as different people, then a subset of the population goes further. And as I was telling you about the first Americans, so the first Americans were there by 15,000 years ago. So this is a very, in 2 minutes, story of how humans moved around the world. What I'm more interested in is what happens when as humans move in different environments. And again, this is very relevant for plants as well and for animals. So what happens when you migrate, when you mix, when you split these populations? How do humans adapt? So why are we all so different? And so when you move in different environments, you can meet different climates, different foods, different parasites. And And this is very important because then we probably have to adapt. And I'll talk a little bit about all these things today. So, you know, we adapted to heat, we adapted to the UV light, we adapted to new pathogens, we even adapted to changing in culture. My PhD was on skin color, and it's a very interesting trait, so human skin color, human skin pigmentation, because it's a visible trait, and it's one of these classic examples of a complex trait, so a trait that is determined by many genes, but which also evolved through natural selection and sexual selection, and it was something that I've always been interested in. And if we talk about UV radiation, you can immediately see how different UV radiations are across the world. So that already tells you something, that if humans would have been moving around, they would have had to adapt to different conditions. So human skin color is determined by the amount and type of melanin, so it's a pigment in the skin. It's like a dark clothes. So basically, if the light comes, the pigment takes the light and doesn't allow it to go in the deeper layers. So UV light, if it goes in deeper layers, can destroy DNA, can destroy all sorts of important molecules. The skin color in native populations correlates very well with the level of UV radiation. So we can do that. We can take a map from NASA. We can see the level of UV radiation in different places, and then we can measure skin color. And what we notice is that it correlates very well. So that already tells us that there's probably been natural selection in response to the UV radiation. And the classic theory is that in the regions with very high UV radiation, for example, Equatorial Africa, there was need for a very high melanin content so that the deeper layers of the skin would have been protected, in particular for the degradation of folates. So against the degradation of the folate vitamins, which are really important for health. But when humans moved towards latitudes with less UV radiation, there was another pressure, and this time what the pressure was for synthesis of vitamin D. So vitamin D is synthesized mainly in the skin, and if you have too much UV, if you have too much melanin, captures the UVB, and there's less vitamin D synthesized. So that's kind of a dual pressure that's existed, and that's our best theory at the moment for differences in skin color around the globe. The genetics of skin color is very complicated. It's a polygenic trait. It has very high heritability, so our constitutive skin color, the one we're born with, it's mainly genetic. It's more than 80% genetic. We know some genes, some polymorphisms that determine some of the variation, but we also know that in the melanosome where melanin is synthesized, there's more than 200 proteins active, so you have around 200 genes and different opportunities for regulation in different places. So basically it's what we call a complex trait. But we know some of these, so there, okay, and I turned it off. Okay, that's good. We know some of these genes. So for example, a polymorphism in this gene, a functional one, we saw that it explains about 30% of the differences between the Europeans and Africans in terms of skin color. One of the most interesting things is that there's been convergent evolution for lighter skin color. So the same polymorphism that exists in Europeans does not exist in East Asians. So if you find a bone of someone from East Asia, you think they're African in terms of their skin color because they have the same genetic variant like Africans, but they are lighter skin. In other words, in their case, there's been another mechanism to get lighter skin. So there's been convergent evolution at least twice for lighter skin, probably 3 times because probably some Neanderthals also had lighter skin. So there have been different instances in our history and evolution when this phenotype evolved. And this shows the frequency of these polymorphisms that I was telling you about. In an unfortunate way, this map shows where with darker, it's the polymorphism that gives lighter skin color. But basically, this is almost fixed in Europe. It's also very present in Central Asia and the Middle East, and it has a patchy presence in India. When I say patchy presence, I mean there's populations with 0% frequency and populations with 100% frequency. Overall, it's about 50. And I'll talk about India because that's where my PhD was. So when I started my PhD, and if there's some students here, I changed a couple of times because the first supervisor, well, I didn't get along with one of the supervisors, so I was looking for a nice supervisor, so that's my advice for all students if they're here. And for supervisors, my advice is for them to be nice to students. But basically, I changed my project and I went to meet my supervisor, and he told me there was a project to study the genetics of skin color in India, so I could go there, collect saliva, talk to people, and I thought that's exactly what works for me, basically. So again, the advice for students is that each of you will be different, and some of you will be better at different things. I prefer to be out in the field more and in the computer than be in the lab. So India is very complex. Why we're very interested in India is because it has this high endogamy. So populations are basically, they have been reproductively isolated for at least 2,000 years. And we know that from genetics. So if you take genetic sequences from different populations, you can see, you can look at them, you can look at mutations, and you know that endogamy has been there for, you see how many generations, you count back, and we know that endogamy ensued about 2,000 years ago. But it has a very high genetic diversity at the same time, and it has very wide phenotypic variation, so it's very diverse in all kinds of phenotypes, including skin color. So that's why it's interesting, and we didn't know much about it. So that was my PhD. I went around, I collected more than 1,000 samples. I was measuring skin color with the derma spectrometer. It's a very simple machine that sends a light on the skin, and then you see a reflectance value, so how much pigment you have in the skin. We collected data mainly from the south, but then we also had genetic data from different populations. And this is, I mean, I'm not going to talk to you much about my results, because that's what people usually ask me, but they say, oh, so what did you find in your PhD? And I'm like, I don't know, I just did my PhD, nothing important. But there were a few important things. So this would be how we look at the phenotype. So this is a distribution of the melanin index, and these are numbers of individuals, and these are several populations from South India. I should say that around 30 or 28, 26 melanin index is what Europeans would usually have in terms of skin color. In Europe, we're not very diverse. It ranges from maybe 20-something to 40, in the Mediterranean, 38. In Africa, we're talking about from between 60 and 90. The darkest recorded melanin index is in the Bungaville Islands, in the Solomon Islands, that's around 110, 120. The interesting thing in India, which we assume but we show, was that there's high diversity. So in only one population, so let's say Timișoara, there were basically individuals who looked almost like Europeans and individuals who would look almost like Africans. So you would have this very high phenotypic diversity, but not in admixed populations. These were not individuals that had just traveled from Europe. It's just been there for a long time. And that's why it's very interesting when you think about traits and phenotypes, what leads to this very huge, very high diversity. We also found some genes, the one I mentioned to you about. We saw that that gene had quite a large effect in this population. We also showed that in India there was not much the UV pattern, but rather the population to which you belonged. So in other words, rather than natural selection, the social forces were more important. Probably one of the most interesting things we found was, and this shows two populations from North India, and why this is very interesting is that these are populations that live nearby, so they live in the same place. So let's say Timișoara with Arad. But their phenotype, their skin color phenotype could not be more different. So you can see the distribution here. They're extremely different. And that for us is really interesting, how diversity has been there for a long time, but has been basically, you have diversity both within but between populations. Another very interesting thing that we found was that you had some individuals here, this is very dark skin, this is basically African in terms of how they look, that had this gene that I mentioned to you about. They were homozygous for the European allele. So these individuals, you could say they had the light skin variant, but they were completely dark. In other words, that makes us think a lot about what a complex trait is. So you have other genetic variants that override the effect of other variants. So, and this is very important for forensics and for other purposes, because if you would have found this individual, you would have said this is a light-skinned individual, but actually they were not, because a phenotype is not made only by one variant, it's made by hundreds of them. And so in different populations, you'll have different genes that override the effect of other genes. And that's a really important point in general for complex traits. Even if we talk about diabetes or other traits, there will be variants that are found in European populations that do something that don't do the same in another population. This is another example of the same idea of traits being very complicated. This is blonde hair in the Solomon Island, and the gene for blonde hair here is a completely different gene than the one in Europe. So it's a completely separate, independent event of blonde hair appearing in the Solomon Islands. And again, this leads us to the idea that traits are really complex and you have different ways to lead to go to the same phenotype. And in the medical world, this should be very important because, as I said, variants that are known to be involved in some diseases will not necessarily be involved in all the individuals depending on their ancestry. This is one of the most important points probably from today. So anyway, let me just look at the watch. So, okay. 5 minutes, do I have 5 minutes? Yeah, okay. There's many different instances of how humans adapted. I'll just go quickly through them. Altitude adaptation is a really interesting one. We have humans that live normally at more than 3,000 meters altitude in the Andes, in the Ethiopian highlands and in Tibet. And they all have been adapting to that. So if you look through their metabolism and to their genes, there are clear genetic signals of adaptation that allows them basically to, with different mechanisms again, independent mechanism in each of these populations. So some populations are better, they have a mutation that is better at capturing the hemoglobin. Other population is better, has dilated blood vessels. So there's been adaptation to high altitude in different populations. A very interesting adaptation that has been recently found is to diving. So in Indonesia, there's people who, the tribe Bajau, who dives a lot, spends a lot of time under the water. They were found to have an enlarged spleen, and there was a genetic variant that was found to be associated with that. So basically, because they've been diving a lot, there was natural selection, there was a benefit for them to have an enlarged spleen. Pathogens are probably the most important drivers of evolution, and we had COVID, and we talked a lot about this. And when we talked about co-host, I think Carmen was talking about co-host, so it's really interesting because now there's a lot of studies to try to understand the evolution and the history of humans in relation to the history of each pathogen. And viruses are probably the most significant force, and there was a study done on human genomes, and they looked at all the non-coding variants, and about a third of all the non-coding variants are associated in the human genome are associated with pathways that fight viruses. So viruses have probably shaped our genome quite a lot. A very interesting example and something I'm really interested in at the moment in Romania is to study the Roma minority group. And in general, we're very interested to study the minority groups or the groups that are recently admixed. And this is a very interesting study that has been done a few years ago by Mihai Neta, a Romanian professor in Netherlands, looking at plague and how humans have recently adapted to plague in Europe. And the idea was very simple. He took a European population, he took a minority Roma population and an Indian population, and the Romas have ancestry from India. And he found some signals in the toll-like receptors that Carmen was mentioning that were changed only in the Romas and the Europeans, so the ones that had been here in the last 500 years. That was basically very fast adaptation to plague that killed around 40% of Europe's population in the 14th century and 15th century. And so basically the ones that survived had some beneficial adaptations. There's been also adaptation to diet, and we see signals in like fatty acids genes, and you can see correlations depending on when humans started farming and when less meat started to be eaten. There's also gene culture coevolution in our history, and the most interesting example is lactose tolerance. Most people in the world are not able to consume milk when they're adults. They don't feel well. However, there's a mutation that appeared only after humans were raising cattle. So it was almost, we changed our culture, and then when the mutation appeared that allowed us to consume milk, that was very beneficial. So that's a very classic example of gene culture coevolution. Interestingly, again, there's different pastoral populations in the world, and they have different mutations, and there's been convergent evolution on this. And I'll just finish by talking a little bit about our cousins, the Neanderthals, and I was talking to you about admixture. I think it's a really interesting example. So when we talk about human diversity and history, we always need to understand that we're all admixed at a certain level. So us in Europe, we're the product of some Paleolithic component, a Neolithic one, and then a more recent steppe component. Neanderthals were our cousins. We are related from about 500,000 years ago. We had split evolutionary. They were around Eurasia, and they were very successful for at least 400,000 years. We always assumed it's a matter of them and us, so they're different. They've been stupid. We killed them. We were very smart as modern humans. But of course, recently we found out that, as they say, make love, not war. We actually met Neanderthals. And it's a very complicated family tree, so we all have about 2% Neanderthal DNA. And we now know that there's been different encounters, so there's been different instances when around 40,000 years ago, modern humans met Neanderthals and they mixed. And that's very relevant, and why I'm telling you that this is that, again, our genomes are the product of different admixture events. We're all mixed. Whether it's recent, like in the case of a Roma minority between Europe and Indian ancestry, whether it's like in Americas now, where people have Native American and European and African ancestry, or whether we talk about some Neanderthal and then modern human ancestry. And this is very relevant in terms of what happens with these genes that we get from people. So usually when we meet with someone, a population is already well adapted in a place, and you take genes, we call it adaptive introgression. You take genes that are usually beneficial. In the case of the Neanderthal, 2% of our 3 billion units of DNA, it's actually quite a lot, but they're not uniformly distributed in our genome. There are places where there's more. And there's many studies being done now on this. So for example, you go through biobanks of human genomes where you know genes and traits, and we found Neanderthal genes being involved in all sorts of things, from depression to mood to immunity. And I was just wanted to mention immunity, and actually a Nobel Prize was received for the Neanderthal genome because it's very relevant for our health today. So it actually seems that we took some genes from the Neanderthals that helped us fight infections when we met them a long time ago, but the same genes that helped us fight infections then now give us an overreactive immune system, which means autoimmune diseases, which means allergies, which means all sorts of things. And I think that this is a very interesting area in medicine. So in other words, a lot of our medical problems come from an evolutionary past. A very interesting recent example was that there was a variant that was predisposing to severe COVID that was actually more present in South Asians, and that variant seems to have was on a stretch of DNA associated with Neanderthals. And I think that's really interesting to think about. And it's the same when we think about diseases today in Europe. As I told you, Europeans are the product of different migrations. The latest big migration that came to Europe was from the steppe. The Yamnaya herders, the Yamnaya, like they were horse riders. What's really interesting about it is that they seem to have brought some genes, again, that were very beneficial in fighting infections, but those genes now that have more frequency in northern populations, predisposed to multiple sclerosis. So I think it's really interesting to always think we have many diseases today that have some origin in our evolutionary past, and that was what I was trying to say. Anyway, thank you so much, and I hope I didn't go too long over time. Thank you.
Vă mulțumim, domnule doctor, pentru interesanta prezentare și pentru faptul că mereu dați scurt invitațiilor la evenimentele științifice din viața universității noastre.
Thank you, Doctor, for your interesting presentation and for always accepting invitation to scientific events in the life of our university.
Mulțumim din nou invitațiilor de onoare pentru prezentările lor valoroase și interesante, care au subliniat faptul că procesul de cercetare este într-o continuă evoluție, accelerat de tranziția către o economie rezilientă, verde și digitală într-o societate inclusivă.
Thank you again to the guest of honor for their valuable and interesting presentation, which highlighted that the research process is constantly evolving, accelerated by the transition toward a resilient, green and digital economy in an inclusive society.
Aici se încheie prima parte a evenimentului de astăzi. Pe ecran este prezentat programul conferinței pe secțiuni, care vor începe la orele 14:30 la nivelul celor 6 facultăți.
This includes the first part of today's event. On the screen is the conference program by section starting at 2:30 p.m. across the 6 faculties.
De asemenea, aș dori să vă mulțumesc cu deosebit respect pentru oportunitatea de a prezenta acest eveniment în calitate de studentă a acestei universități de prestigiu la nivel național și internațional.
I would also like to thank you with great respect for the opportunity to present this event as a student of this nationally and internationally prestigious university.
Vă mulțumim tuturor pentru participare și vă dorim succes la prezentările conferinței.
Thank you all for your participation and we wish you success in your conference presentations.